Good morning everyone. Today my topic is mutation. Firstly, mutation is a change in the DNA sequencing of the organism. For example, it is a change in the composition of the DNA, RNA, nuclear, their development, function, reproduction and other things. There are different type of mutation. Firstly, based on the scale. Firstly is gene mutation, or we can say it point mutation. It involves change in the single nucleoside or a small change in the nucleoside of a DNA. There are some common type substitutaneous. One base pair is replaced by another. There are further classification in the transition a pure replication another purine or pyrimidine replace another pyrimidine. There can be few steps transition, invention and detention. Another one is frame shift mutation. Involve shifting of one frame or replacement or the deletion of the one chromosome of a DNA. Next mutation type is chromosomal mutation. Affect a large segment of DNA or entire chromosome. There are common type for detected one a portion of the chromosome is loose. Duplication there is a new duplicate form of a DNA. Invention a segment of chromosome is reversible end to end. Translocation segment from two different chromosomes are exchanged. Another mutation is spontaneous mutation, occur naturally during DNA replication, repairing or recombination process. This mutation can lead to a cause of error made by a DNA polymerase. Another one is induced mutation, caused by the external factor known as mutation. Mutation can be physical, chemical, biological in nature. Physical mutation involve radiation. Chemical include certain chemical like excreting agent, intracatalytic agent, or base enzymes that affect the DNA structure. And biological mutation include various viruses or transport element can be insert themselves into the genome. Based on the effect of protein function, there are different mutations. First, silent mutation. Change in the DNA sequence without affecting the amino acid sequence. Another was miscellaneous mutation, a change in a single nucleotide that resulting in the substitution of the one amino acid to the another in the protein. Nonsense mutation, a mutation that convert a code from an amino acid into a stop codon, leading to a premature temperament of the protein. Another one is frame shift mutation, we have already discussed. Now let's discuss the mechanism of DNA repair. Firstly, cell has a mechanism to detect and repair mutation to maintain genomical interaction. Some key repair mechanisms involve mismatch repair, base extinction repair, UV radiation, non-hologeneal and joining. Let's discuss these in detail. First is mismatch repair. In which a fixed mismatch base pair that were incorrectly incorporated during DNA replication. Base extinction repair repairs single based alternating like deamination of the oxidative. Nucleotide extinction repair remove badly DNA detected such as those caused by the UV radiation. Homologous recombination repair double stranded structure of a DNA. Now let's discuss about some disease in the mutation. First is single gene disorder, which is cystic fibrosis caused by the mutation of CFTR gene leading to a malfunctioning chromatin chain and building up of a thick mucus in the lung. Sickle cell anemia caused by a mismatch mutation in the HBB gene resulting in the abnormal hemocytic protein. There can be a cancer and many more diseases. In last, I want to conclude that mutation is can be reversible or maybe irreversible in many cases. My another topic is regulation of gene. Gene regulation refers to the complex process by which cell control the examination and timing of the gene to ensure that the correct protein are made at the right time, in the right amount and the right cell. The regulation is essential for the purpose function of all the living organisms for bacteria and human and allowing a cellular distribution, reporting on the environmental change and maintaining of a homeostatic. Here is a detailed view. Firstly, level of gene regulation. Gene regulation can occur in various steps of gene expression from DNA to functional protein. Firstly, transcriptional gene. This is the most important and common level of gene regulation. It involves some steps. First, promoter, a specific DNA sequence where RNA polymerase bind to the initiate transmitter. Transmit factor, protein that bind to a specific DNA sequence near the gene promoter to increase activity or decreasing repair transmission. Enhancer and silence, epidermal modification, post-transcription regulation, RNA sprinting, RNA stability, RNA ending, and many more, like transcript regulation, post-transcript regulation. Now let's discuss about the prokaryotic and eukaryotic gene regulation. Prokaryotic gene regulation, such as bacteria, regulate their gene in a response to environmental change to maximum efficiency. One of the example is E. coli, which control the breakdown of lactose. Eukaryote gene regulation in the prokaryote, eukaryote cells have a more complex gene regulation system due to their multicellular nature and need for a cellular definition. Eukaryote use mechanisms like chromatin recombinant, epigenetic regulation, etc. Some example of gene regulation system are lake operon, where the lake operon of E. coli is well studied example for the transcription regulated in response to the environmental condition in a presence of a lactose. The operation is turned on, allowing the bacteria to mineralize in the sugar. Hox gene, eukaryotic Hox gene are the critical for detecting the body plan on the organism. These are regulated by the highly specific manner during early development and ensuring the body part for the correct place. Gene regulation and disease. There are different disease caused by these. Firstly, cancer. Genetic cancer is one of the most critical disorder. Then in gene disorder, some of the gene disorder are mutation in the regulatory element of the beta glucose gene can lead to the disease like beta thyroxine. Thank you.